So fundamentally, we know that engineered T cells can attack and kill cancer with significant efficacy. We also know that target selection is critically important. So not every cancer is defined by markers that are easy to go after. So terminally differentiated B-cell malignancies like myeloma and B-cell lymphoma have markers on them like CD19, which are present on normal tissue that can be injured and grow back in a way that's safe, and those proteins easily define those cancers. In solid malignancies, we have many fewer markers. Solid malignancy and human tissue is much more similar in ways that are problematic. So the initial efforts at CAR T therapy, second generation CAR T therapy against HER2, which is a very well established oncologic marker, were pretty dangerous. People died, and there was significant injury, in particular on the lungs, which is a known toxicity with HER2-directed therapies. So the TAC T approach is a way of saying we understand and recognize that T-cell therapy can treat cancer, but that we can't just use a CAR T. We need a T-cell that has the ability to turn off and turn on in a more regulated way and not generate that on-target normal tissue effect that a conventional CAR T does. And so the TAC Ts contain a normal T-cell receptor complex with all of its upregulation and downregulation, and then there's a modification that promotes survival that engages the T-cell receptor complex without constitutively activating it, and then is bound to an extracellular moiety, which can then go to a target, in this case HER2. And if the platform is as safe and effective as we believe it is, it's not just HER2, it's many different targets, and you could just name all of them. Things that are a little bit too risky for a CAR T become a possibility for a T-cell activating complex.